Good afternoon. I'd like to welcome all of our virtual attendees for today's Science of Childhood Cancer seminar. All right, let's go ahead and get started. So welcome everyone. I'm Suzanne Baker from St. Jude Children's Research Hospital, and I'm really delighted to host today Dr. Michelle Manji from Stanford University for our Science of Childhood Cancer seminar series. We are going to have a terrific lecture from Dr. Manji, and she'll take some questions at the end of the lecture, but she has also graciously agreed to stay on for an extra 15 to 30 minutes after the hour for additional questions if people have more questions at the end of her seminar. So now it's my great pleasure to introduce Dr. Michelle Manji. Michelle did her MD PhD degrees at Stanford University with her PhD work under the mentorship of Theo Palmer. So she was working in sort of neuroprogenitor cell research from a very early stage in her training career. She did a residency at Harvard and then a fellowship back at Stanford, where the research component of that was done with Phil Beachy. So Dr. Manji is truly a clinician scientist. She's board certified in both neurology and neuro-oncology. She is and has been on the faculty at Stanford since finishing her fellowship there. So I think Michelle is an exceptional scientist. She has put together a really unique and innovative research program using a very sophisticated consideration of the tumor microenvironment and building on her strong background in neurodevelopment. So she has really, I think, created this major exciting field at the moment of how neuronal glial interactions can actually promote tumor development and tumor progression. She has done this through multiple technical approaches, including biochemical purification of secreted factors from neurons that are growth promoting for tumors, an in vivo demonstration of neuronal glial interactions, neuronal tumor interactions, and how they affect this important and interacting microenvironment. Importantly, she has gone on to take this information, show that it can be leveraged to improve therapeutic approaches for pediatric brain tumors. So extremely exciting and innovative work. Michelle's work has been recognized by a number of awards, including the Jerome Posner Neuro-Oncology Investigator Award from the American Academy of Neurology. She received a National Institutes of Health Directors Pioneer Award, a Presidential Early Career Award in Science and Engineering, and she's an endowed faculty scholar in the Biomedical Innovation Initiative at Stanford University, among other awards. So I'm really looking forward to this talk today. I'm not going to take up any more of Michelle's time. So welcome, Michelle. We look forward to hearing all about your work.
Thank you so much for that wonderful introduction. And it's so wonderful to be here with you, at least virtually. So thank you so much for the invitation. I'm really excited to talk to everyone afterwards. As Dr. Baker said, I am a pediatric neuro-oncologist really focused on better understanding the developmental origins and therapeutic opportunities for childhood high-grade malignancies, particularly diffuse midline gliomas like diffuse intrinsic pontine glioma. And I want to tell you one story today in two parts about the way in which neurons interact with the glial cells that in the healthy brain form the myelin sheath in a process that we're increasingly understanding can be plastic and adaptable to experience and that supports normal neural circuit and cognitive functions, and how these really powerful interactions can be subverted and hijacked in the context of glial malignancies. But before beginning anything, I like to take kind of a broad consideration of neural development. And I find that the very best way to do that is by showing you pictures of my children. So this is our youngest daughter, Sophie. And by the time we had Sophie, we realized that, there was just so much neurodevelopment to accomplish each day in human infancy that we made her a onesie with a checklist so she could kind of keep track of everything. And it really, when you take a step back and consider it, human neurodevelopment is astounding, and human infants undergo just enormous strides in development in the postnatal period. For example, in the first year of life, each day, human infants make something on the order of 500 billion synapses. 14,000 new hippocampal neurons, 127 million cerebellar granule cell neurons. And as impressive as all of that is, perhaps the most onerous task that we have in the postnatal period is to myelinate our central nervous system. We are a semi-altricial species. We are born almost unable to control much movement. And then there's this just incredible amount of development over the first year, much of which really is in thanks to this process of myelination. And myelination of the central nervous system is actually a process that in the human central nervous system extends over more than 3 decades of life. So this is a very long developmental process, relatively longer in humans than even in other vertebrates and in particular other non-human primates. And, this process of developmental myelination follows very predictable topographical and chronological patterns. There's a spatiotemporal pattern to this developmental process. We're born with almost no myelin in the central nervous system, and in the perinatal period, myelination begins kind of in the center of the brain, around the central sulcus, and then progresses towards the poles. Similarly, it begins in the rostral spinal cord and then progresses down the spinal cord. And actually, in the first year of life, you can observe the motor milestones attained as really a demonstration of the stage of myelination that is achieved. So first, human infants are able to raise their head, then bring their hands together. By about 6 months, they can sit up as myelination progresses down to the thoracic cord. And by about a year, typically can walk as the myelination progresses down to the lumbosacral cord. And in general, more basic circuitry, like the circuits that underlie motor and sensory behavior, myelinate before the neural circuits that underlie more complex cognitive functions. And so, for example, there's a discrete wave of developmental myelination in mid-childhood in the brainstem, particularly in the corticospinal tracts. And later in adolescence and young adulthood, there's active myelination in the cortex and subcortical association fibers. This process of myelination of new oligodendrocytes that are generated by oligodendrocyte precursor cells throughout childhood, adolescence, and we understand throughout adulthood. This process of making new oligodendrocytes, these oligodendrocytes then ensheath the axonal membrane to decrease transverse capacitance of the membrane and increase the speed of neural impulse conduction down the axon. This is something that even after development is complete continues. And we've actually recognized from historical work of pioneering pathologists for more than 100 years that there's this accumulation of myelin, particularly in the cortex and in the subcortical fibers. You can see that here in this lithograph showing you the increase in this black stained myelin over the lifespan, increasing in the human cortex. And in more recent studies in rodents leveraging newer in vivo imaging techniques, it's very clear that there's an accumulation of myelin, not just myelin turnover. Oligodendrocytes are actually very stable cells, but rather an accumulation of myelin in parts of the nervous system that are not completely or not ideally myelinated. And indeed, there is quite a lot of available axonal territory in the cortex and subcortical fibers throughout life, sort of suggesting that there may be some inherent tunability to the myelinated infrastructure. And this is, intrinsically fascinating, but also really important as we consider that gliomas of childhood also arise in a very predictable spatiotemporal pattern. And in fact, in a pattern that maps well onto this pattern of developmental myelination, so that at a time when there is a particularly robust wave of myelination in the ventral pons, this is when one of the worst human cancers, diffuse intrinsic pontine glioma, tends to occur. And similarly, at a time when there's fairly active myelination in the cortex and subcortical fibers in adolescence and young adulthood, this is when we tend to see hemispheric high-grade gliomas of childhood or pediatric glioblastomas. And this is concordant with observations from my lab and from others that many of these high-grade gliomas may indeed arise from precursor cells in the oligodendroglial lineage, either oligodendrocyte precursor cells or just earlier pre-OPCs. And certainly fits with molecular observations about many forms of high-grade glioma, and in particular the diffuse midline gliomas, that the cancer cells very closely resemble normal OPCs. And so we may glean really important clues to gliomagenesis by better understanding the normal process of gliogenesis. And so that raises the very basic question: what does regulate the proliferation and functional differentiation of oligodendroglial lineage cells? Well, one idea that has been in the literature for some time, actually first introduced in the 90s by Ben Barris when he was a postdoctoral fellow with Martin Raff, was the idea that neurons themselves may regulate the extent to which their axons are myelinated, and that this might happen in an activity-dependent manner. There were a number of studies in the 2000s that really nicely supported this hypothesis, really nice in vitro studies and correlational studies, but it remained a pretty hotly debated topic, a pretty controversial topic in the myelin field, because there are also very clearly activity-independent modes of myelination. We know that oligodendrocytes will, in a petri dish, myelinate fibers of the appropriate diameter, and so it's very clear that there are activity-independent modes, at least as well. And so when I started my lab, now I guess about 9 years ago, one of the first questions that we set out to answer was that, does neuronal activity regulate myelination in vivo in awake behaving animals? Can myelin be plastic and adaptable? And so I want to focus briefly this first part of the talk on this idea of myelin plasticity, of activity-dependent adaptive changes in the myelinated infrastructure of the brain. And I want to introduce you, of course, to the people in the laboratory who did this work. This is Erin Gibson, who was my very first postdoctoral fellow. She has recently started her own laboratory at Stanford in the Department of Psychiatry, and she's definitely somebody to watch. She really led this effort in the lab from the very beginning. And this is Anna Garrity, who is currently a senior postdoctoral fellow in the lab. And what Anna and Erin and others in the lab have done is to leverage the neuroscience technique of in vivo optogenetics to ask this question. So as many people in the audience probably know, optogenetics is a technique that allows for light-based control of target populations of cells and control of, in the case of neurons, action potential. So these are microbial light-sensitive opsins that are isolated from algae and other forms of single-celled organisms. This is channel rhodopsin 2, which is an algal light-sensitive blue light-sensitive cation channel. And if you express channel rhodopsin 2 in cortical projection neurons in the mouse cortex, you can then stimulate neuronal action potentials with light delivered at the surface. And if you do this in the motor planning area here of this mouse that expresses channel rhodopsin 2 in its cortical projection neurons, and you shine the light at a frequency that's commensurate with the frequency at which these neurons typically fire, in this case 20 hertz, then the mouse begins to execute complex motor output. So we know that we have, in a physiomimetic way, recruited circuit activity. We can identically manipulate and deliver light to wild-type littermate controls that don't express the opsin, and there's no behavioral output. So we use those animals to control for light and surgery, et cetera. And then after light stimulation, we can ask very straightforward questions about, how other cell types within this known circuit respond to changes in neuronal activity. And what we found is that when we stimulate the premotor circuit in mice, that there is a rapid and robust circuit-specific response of oligodendroglial precursor cells. There's within just 30 minutes, we find that there is quite a bit of proliferation, very specifically within the corticocallosal projections of the premotor circuit, and not the corticofugal projection, suggesting that different subtypes of cortical projection neurons might differentially interact with myelin-forming cells. If we fate map these cells over time, we can deliver the thymidine analog, EDU, and then watch what the cells that divide at the time that we optogenetically stimulate the animals become over the subsequent few weeks. And we find that there is indeed an increase in newly generated oligodendrocytes and a change in the ultrastructure of myelin within that circuit after daily stimulations over the course of a week. Those changes would be predicted to increase the speed of neural impulse conduction, to change circuit dynamics, and potentially to influence behavior. And what we found was that indeed there was an improvement in motor functional behavior that depended upon the generation of these new oligodendrocytes. So a next set of questions are what molecular mechanisms are mediating this interaction? And one leading molecular hypothesis was that activity-dependent secretion of the neurotrophin, brain-derived neurotrophic factor, signaling to its receptor NTRK2 or TRKB on the oligodendrocyte precursor cell might be an important mechanism. And so we tested this hypothesis using two different genetic mouse models. One mouse model made by Mike Greenberg's lab at Harvard, in which there are baseline levels of BDNF, but the mouse has had promoter 4 Krebs binding site deleted, such that there is no increase in BDNF expression or secretion in response to neuronal activity. And in a second mouse model, we instead conditionally and inducibly deleted the TRKB receptor just from oligodendrocyte precursor cells at various points of postnatal development. And what we found is that if we optogenetically stimulated premotor circuit activity in either of these genetic mouse models, that the activity-regulated response was completely lost. In mice that either have loss of activity-regulated BDNF ligand secretion or OPC-specific expression of the BDNF receptor, there is no increase in proliferation in response to neuronal activity in comparison to their wild-type littermate controls. As there's no OPC proliferation, there's also no increase in activity-regulated new oligodendrocyte generation, and accordingly no change in myelin ultrastructure in either mice. And so while certainly this mechanism is more complex than just this one interaction, BDNF to TRKB signaling is one necessary component of cortical projection neuronal activity regulated myelin plasticity. And that gives us a molecular handle to begin to ask what role adaptive myelination may play in various forms of neurological and in particular cognitive function. And so I'm going to show you the results from one set of these kinds of experiments, testing attention and very short-term memory function. So this is a pretty straightforward behavioral test called the novel object recognition test. In this, we put a mouse in a chamber with two identical objects. We allow the mouse to explore and get to know these objects, and then we remove the mouse from the chamber for just a short period of time, just 5 minutes. And then replace the mouse in the chamber with one object being replaced with a novel object. Now, if the mouse had paid attention to and remembered what it had previously explored, it would spend a little bit more time with the novel object. And that's what we see in healthy, intact mice. In mice that have lost the ability to respond to neuronal activity-regulated signals in their oligodendroglial lineage, in OPC-specific TRKB knockout mice, we find that when the TRKB receptor is deleted either in adolescence or in young adulthood, that there is a sort of failure of performance in this test. In comparison to their littermate controls, mice that have lost the ability to respond to activity-regulated signals in their myelin spend equal time with the novel and the familiar object. And so this loss of activity-dependent myelination we're finding, which just to summarize, in cortical projection, neurons depends upon communication between neurons and OPCs through BDNF to TRKB signaling, generation of new oligodendrocytes, changes in myelin ultrastructure that we are increasingly recognizing contribute to coordinated network function and therefore intact cognitive behavioral performance. After chemotherapy, like systemic high-dose methotrexate, there is a complete failure of the adaptive response. And that failure of adaptive myelination actually depends upon direct activation of methotrexate on microglia. Microglia, in their neurotoxic state and through mechanisms we have yet to fully understand, induce a loss of BDNF expression in cortical projection neurons. And if we deplete microglia, this actually rescues BDNF expression in cortical projection neurons and also rescues behavior. And so this loss of activity-dependent plasticity appears to contribute importantly to the impaired cognitive performance that mice in this preclinical model, and we think children experience after exposure to systemic chemotherapy like methotrexate. And so, how do these new oligodendrocytes integrate into the myelinated infrastructure? Is it that the new oligodendrocytes remodel internodes in existing myelinated axons? Is it that they myelinate previously naked axons? Or is it that they myelinate previously unmyelinated segments of axons? Some really beautiful work out of Paula Arlada's lab recently has shown that in the cortex in particular, if you follow a single axon, there's intermittent myelination. So lots of opportunity for a new oligodendrocyte. The conduction velocity a little bit and potentially allowing for adaptive tuning of circuits. What is clearly emerging is that all three of these mechanisms can and do happen in different parts of the brain, and that the addition of these new myelin internodes or remodeling of myelin to change the geometric parameters in a way that changes the speed of impulse conduction, this promotes in the healthy brain coordinated circuit function like oscillatory synchrony. And accordingly, as adaptive changes in myelin seem important for supporting normal and coordinated circuit function, we and others are finding lots of implications for myelin plasticity in a variety of cognitive functions. So Bill Richardson's group in the UK has shown really clearly that there is a role for new oligodendrocytes in motor learning. We've found a role for adaptive oligodendrogenesis and myelin changes specifically in attention and short-term memory, and in collaboration with Paul Franklin's group in Toronto, also in memory consolidation. So there are a number of implications then for myelin plasticity in disease. And one is to leverage what we're learning about myelin plasticity to promote remyelination after demyelinating injury. There's also the potential that loss of adaptive myelination or dysregulation of adaptive myelination may importantly contribute to a range of different kinds of neurological diseases. We've found evidence in chemotherapy-induced cognitive impairment that loss of myelin plasticity through cancer therapy-induced microglial activation as sort of the central mechanism is a really important and potentially targetable mechanism underlying impaired attention and other forms of cognitive dysfunction. In emerging work from the lab, this promotion of oscillatory synchrony that myelin plasticity seems to promote, we're finding plays an important role in diseases of hypersynchrony, like childhood absence epilepsy. And so Juliette Knowles in my lab, who's a pediatric epileptologist, together with John Huguenard, and I have recently found that the aberrantly increased myelination that happens in the context of absence epilepsy also then contributes to increasing seizure severity. And given these robust proliferative influences of neuronal activity on normal glial precursor cells, it certainly stands to reason that there may be a similar signaling relationship in malignant glioma. And so I want to focus the second half of the talk on this idea of malignant myelin plasticity, activity-dependent glioma growth. And of course, to introduce you to the people in the lab who have been working very hard on this question. This is work that was initially in the lab led by Hamsa Venkatesh. She was then a cancer biology PhD student and then subsequently a postdoctoral fellow in my lab together with Rob Malenka's lab. And she's now just about to begin her own independent laboratory at Harvard Medical School, and definitely somebody to keep an eye on. She's absolutely brilliant. More recently, Ewan Pan is a senior postdoctoral fellow in the lab who has been studying neuron glioma interactions in low-grade glioma. And this is Tara Barron, who's a fairly recently joined postdoctoral fellow. She is an electrophysiologist by training and has been contributing importantly to some of our more recent work. And as this audience knows well, this is this sort of spectrum of malignant gliomas that we think hard about. These are MRI images and showing these terrible diseases. This is diffuse intrinsic pontine glioma, very closely related. We understand from Dr. Baker's work, really pioneering work in this disease, very closely related disease called diffuse midline glioma of the thalamus. This is a pediatric cortical glioblastoma that is histone wild type. And this is apparently similar, but we now understand really molecularly distinct disease called adult glioblastoma. And one thing I want to point out is in these MRI scans is that these are not discrete masses. These are cancers that diffusely integrate and invade normal neural tissue, normal neural circuits. And in fact, we see a circuit-based pattern of invasion in many cases. It's not unusual to see diffuse intrinsic pontine glioma invade and spread apparently along a circuit like the limbic circuit. And so we wondered, does neuronal activity promote glioma proliferation? And this is a question that really, I think our neuropathologists have recognized as a potentially important interaction for many, many years, because this phenomenon of perineuronal satellocytosis, the propensity of high-grade glioma cells to cluster in very tight microanatomical association around mature neurons in the microenvironment really suggests that there's an important signaling relationship. And I want to just say parenthetically that this particular H&E image comes from an autopsy specimen from a 5-year-old patient of mine. And it was actually his donation that allowed us to generate one of the first patient-derived cell cultures and xenograft models of diffuse intrinsic pontine glioma. And it's really through such tissue donations from patients and their families that we and you at St. Jude and many others have been able to make progress in this terrible disease. And so I just want to acknowledge that critically important contribution from patients. And so to ask whether neurons are influencing glioma growth in the childhood brain, we turn to the same experimental model system that we previously used to look at normal neuron glial interactions, but this time in the context of a diffusely infiltrating childhood high-grade glioma. So we're stimulating the frontal cortex here, and so we used a cortical glioblastoma from an adolescent patient of mine. And what we found is that when we optogenetically stimulate cortical projection neuronal activity, that just like their normal counterparts, the malignant glioma cells increase their rate of proliferation, and that this results in an increase in tumor burden specifically within the stimulated circuit. So brain activity can influence glial malignancy growth. And we wanted to understand the mechanisms by which this may be occurring. And so we turned to a different model system, healthy acute cortical slices from mice, non-tumor bearing brains. And then we collected the secreted factors from these acute cortical slices with varying levels of activity. So either we collected conditioned medium from slices that had baseline spontaneous activity, or we optogenetically stimulated neuronal activity. And in another set of experiments, we tetrodotoxin silenced activity using this voltage-sensitive sodium channel blocker tetrodotoxin. And what we found is that when we took the conditioned medium containing secreted factors from these brain slices and placed them onto patient-derived cultures of high-grade glioma, that there was an activity dose-dependent increase in glioma cell proliferation that's lost when the brain slices were silenced with tetrodotoxin. Now, as we know, these different high-grade gliomas of childhood and adulthood are really clinically and also importantly molecularly distinct diseases. But we find that the response to activity-regulated secreted factors is conserved across all these different kinds of high-grade glioma, from pediatric from histone wild-type pediatric glioblastoma to diffuse intrinsic pontine glioma, adult glioblastoma, and anaplastic oligodendroglioma. And so we wanted to understand what was in that conditioned medium. And so to do that, we did some very basic biochemistry, separated molecules based on molecular size. We found that the active fractions was in the macromolecular fraction. It was sensitive to heat, insensitive to DNase, RNA, so likely to be a protein or group of proteins. We then did a variety of proteomic analyses of the conditioned medium, and that generated this list of differentially expressed proteins as a result of neuronal activity. And so from this list, we then did candidate sufficiency and necessity testing. And what we found was that there were two key glioma mitogens. Not unexpectedly, as BDNF is an important molecular mediator of normal neuron-glial interactions, we found that indeed BDNF stimulates the proliferation of a variety of high-grade gliomas, including DIPG and pediatric glioblastoma. But really unexpectedly, we found this molecule called neuroligin 3. Now, neuroligin 3 functions is a very robust glioma mitogen across different molecular subtypes of glioma. And it's a really well-known protein. Neuroligin 3 is a postsynaptic adhesion molecule. It canonically binds to presynaptic norexin. It was discovered by Tom Sudoff and has been very, very closely studied because in one particular mutant form, it contributes to a familial form of autism. It's present at both excitatory and inhibitory synapses and contributes to regulating synaptic strength. But it was not known to be a mitogen in any context. And actually, it was not even known to be secreted. But we find that neuroligin 3 is cleaved at the membrane, and then this large N-terminal ectodomain is shed in a strictly activity-dependent fashion through the enzymatic activity of a metalloprotease called ADAM10. So the next basic question is, what cell types are shedding neuroligin 3? There are many forms of neurons, each of which can participate in synapses and be a postsynaptic partner. But also oligodendrocyte precursor cells are a postsynaptic cell type. These so-called axon glial synapses were first discovered by Dwight Burgles in the early 2000s and then very well characterized over the last 20 years. We still don't actually know what functional role they play in myelin biology, but they're present on almost every OPC, and they're very clearly electrophysiologically functional as well. OPCs express high levels of neuroligin 3. And so to test the relative contribution of neuroligin 3 shedding from different cell types, we genetically inducibly deleted neuroligin 3 from either a subtype of neurons or from oligodendrocyte precursor cells. And what we found was that not only neurons, but really quite importantly, OPCs contribute to this secreted pool of neuroligin 3 that is then available in the tumor microenvironment. So how important is neuroligin 3 to high-grade glioma growth? There are many cell intrinsic mechanisms by which a glioma cell will grow, multiple microenvironmental mechanisms. In fact, we just discussed two. And so we didn't know how relatively important this might be. And to test that, we xenografted patient-derived high-grade glioma cells into the environment of either the wild type or the neuroligin 3 knockout brain. And I'm showing you here data from that histone wild type pediatric cortical glioblastoma. Really unexpectedly, we found that loss of neuroligin 3 from the microenvironment didn't just slow growth, it it caused an absolute stagnation of growth, and that this was true for up to 6 months. If we monitor these tumors using in vivo bioluminescent imaging over time, we see that in contrast to the tumors that are in the neuroligin-3 wild type environment that grow exponentially, that there's just this stagnation of the tumors in the neuroligin-3 knockout background until about 6 months, at which point a subset of these tumors find a way to circumvent this apparent complete dependency and grow through mechanisms that we're still working hard to understand. This dependency on neuroligin-3 in the microenvironment extends across different brain regions. So here I'm showing you DIPG in the mouse pons, growing in the wild type pons, not growing in the neuroligin-3 knockout pons. We see this dependency in patient-derived models of DIPG as well as adult glioblastoma, but this dependency does not extend to patient-derived models of breast cancer brain metastases, suggesting that while this is an important mechanism for many forms of high-grade glioma, it may not extend to all forms of brain cancer. So I've just told you that neuroligin-3 is this really interesting therapeutic target, and also that ADAM10 mediates its cleavage and release into the tumor microenvironment. And so we tested the idea that a systemic ADAM10 inhibitor that conveniently enough is fairly brain penetrant might phenocopy the loss of neuroligin-3 in the tumor microenvironment. And we were very happy to see that that was in fact the case in our preclinical models. There's quite a stagnation of tumor growth in various forms of high-grade glioma preclinical models in response to this ADAM10 inhibitor. So I'm very pleased to say that we've just recently opened a clinical trial of this ADAM10 inhibitor within the Pediatric Brain Tumor Consortium, and fingers crossed that we see some similar degree of benefit in children with high-grade gliomas as we do in these mice. So just to recap what I've just told you, neuronal activity results in the cleavage and shedding of this postsynaptic adhesion molecule neuroligin-3 through the enzymatic activity of ADAM10. ADAM10 is actually released from the presynaptic neuron and we think in synaptic vesicles. The shed neuroligin-3 then binds to an as of yet unidentified binding partner on the glioma cell. We're working hard to identify that binding partner. We do not think it is the canonical neurexin. And then there's a rapid and robust stimulation of multiple oncogenic signaling pathways. So within 5 minutes, we see increased focal adhesion kinase activity, and downstream of focal adhesion kinase, we see stimulation of SRC, RAS, and PI3 kinase mTOR pathway activity. So that helps to explain the role of neuroligin-3 as a mitogen, but it doesn't explain this dependency. Why can't the tumor cells progress in the absence of neuroligin-3 in the microenvironment? So we dug a little bit deeper and we looked at the gene expression changes that are attributable to neuroligin-3 exposure in the glioma cells. And what we found, kind of surprisingly, was a number of different synapse-related genes. There's a feed-forward effect of neuroligin-3 on its own expression in glioma, together with an upregulation of the BDNF receptor tract B, but also a number of glutamate receptor subunits and other synapse-associated structural proteins. If we look at that in a different way, using single-cell transcriptomic data from Mario Suva and Mariella Philpin's groups, we can look at the expression of these synapse-associated genes across the major classes of high-grade glioma, from H3K27M mutant midline gliomas to IDH wild type and mutant adult glioblastomas. And similarly, in these primary biopsy samples, we see very robust glutamate receptor and other synapse-associated structural protein expression. And if we look at that single-cell transcriptomic data in a different way, looking at each tumor, we see that there's a subpopulation of cells with synaptic gene enrichment. And perhaps not surprisingly, the synapse-associated gene enrichment is found in the OPC-like compartment of the tumors. So that raises kind of a crazy question. Like there are axon glial synapses, might there also be axon glioma synapses? Well, when we look at primary biopsy samples by electron microscopy, we do in fact see quite a number of synaptic structures. But we can't in those primary samples tell whether the participating cells are normal or malignant cells. If we look, however, in our patient-derived xenograft models in which we can express something like green fluorescent protein in the malignant cells, then we can do immunoelectron microscopy and look to see which cell types are participating in these synapses. And what we find is that on about 10% of the glioma processes, there is indeed a synapse that we can identify with very clear presynaptic mouse neuron axon terminals, and then a postsynaptic glioma cell with a very clear postsynaptic density. You can even see the presynaptic vesicles here and here. Looking at those structural synapses is a different way to ask the question of whether neuroligin 3 is indeed promoting structural synaptogenesis. We find that if we co-culture glioma cells together with either neuroligin 3 wild type or knockout neurons, that there are many fewer structural synapses that form in the absence of microenvironmental neuroligin 3. But are these synapses functional, or is this just kind of a shadow of the cell type from which we think these tumors arise? To test that, we turn to a very well-known and tractable circuit, the hippocampus, and we xenografted DIPG cells labeled with green fluorescent protein into the CA1 region of the hippocampus. We then, after allowing these cells to engraft for about 8 weeks, made acute hippocampal slices, and here's a picture of one of those. You can very clearly see the GFP positive cells. And then performed whole cell patch clamp electrophysiology. Here you see a glass pipette going into a GFP positive glioma cell. And when we do that, we can record from the glioma cell while stimulating the axonal afferents into the CA1 region. And then after we record, we can fill the cell that we were recording from with biocytin dye to confirm that the cell that we recorded from was indeed a malignant cell. And when we do this and record from the glioma cells, what we find is that there are indeed postsynaptic, excitatory postsynaptic currents in the cancer cells. We see this in about 5 to 10% of the cells that we patch. These currents are blocked by tetrodotoxin, so that means that they're dependent upon neuronal action potentials. They exhibit multiple electrophysiological characteristics of a true synapse, like paired pulse facilitation and single vesicle events called mini EPSCs. More specifically, these glioma EPSCs are AMPA receptor dependent, and even more specifically, they're calcium permeable AMPA receptor dependent. Now, in a different subset of glioma cells, rather than a synaptic current, we instead saw a very different electrophysiological response. So in a subpopulation in each tumor, sometimes representing upwards of 60% of the cells in the tumor, we saw these prolonged currents that scaled with the field potential, meaning that the more neurons were active, the longer and larger these currents were. And what we determined that these represent is potassium evoked currents. So these, as we know, when neurons fire action potentials, there's an increase in extracellular potassium concentration. This triggers these potassium evoked prolonged currents. And we noticed that in the cells that exhibit these prolonged currents in response to neuronal activity, when we dye filled them, the dye didn't stay typically in just one cell, but rather diffused into a network of glioma cells. And so that called to mind observations in adult glioblastoma that gap junctions can couple glioma cells into an interconnected network. And we wondered whether, in this case, gap junctions were coupling DIPG cells in an electrically coupled network. And in that case, if perhaps this gap junction coupling might be contributing to amplifying the current that we were observing. And so we blocked gap junctions using a couple of different methods. I'm showing you data here from a migraine medicine called meclofenamate that blocks gap junctions. And indeed, what we found is that with meclofenamate application, the current really decreased in amplitude. And we saw this across multiple different kinds of gap junction blockers, suggesting that gap junctions are functioning in the cancer to amplify these potassium evoked currents. So we can visualize this electrical activity in the cancer cells using genetically encoded calcium indicators. When a membrane depolarizes, there's often a calcium transient, and so we can visualize these here in DIPG cells xenografted into the hippocampus through 2-photon microscopy. And you see that when we stimulate the Schaefer collateral axonal afferents, that evokes a calcium transient. If we were to apply a bath applied to trodotoxin, that blocks these calcium transients. And of course, there's spontaneous activity in the tumor, which for whatever reason, my movie is, I'm going to try to restart this because it's a really good movie. For some reason is not playing well here for us. Too bad. In any case, what we find is that, this cancer is really an electrically active tissue, and we have not been approaching it as such. And if this is a tumor that undergoes rapid and frequent membrane depolarization, which is a very metabolically expensive thing to do, there might be a functional significance of this for the cancer, and that actually makes a lot of sense from the perspective of brain development. We know that in the developing brain prenatally, that membrane depolarization seems to regulate normal neural stem and precursor cells in very important ways. And so it may be that membrane depolarization alone is promoting proliferation in glioma. And so we tested that again using optogenetics, but this time using the channel rhodopsin 2 expressed not in the microenvironmental neurons, but rather in the tumor cells themselves. And so if we express channel rhodopsin 2 in a glioma cell, like a DIPG cell, and we shine blue light, we do then see these light-evoked depolarizing currents. And so then we can xenograph the tumor to the cortex and stimulate membrane depolarization in the tumor optogenetically. We find that when we do that, that there is indeed an increase in glioma cell proliferation. Conversely, if we block neuron to glioma glutamatergic neurotransmission using expression of a dominant negative version of the GluA2 subunit of the AMPA receptor, or pharmacologically using AMPA receptor blocking anti-epileptic drugs, we find that there's a stark inhibition of growth. Conversely, we can overexpress wild-type GluA2. We find that actually accelerates tumor growth and decreases survival in mouse models. And if we instead overexpress the dominant negative version of the GluA2 subunit, that extends survival in a particularly aggressive model of patient-derived DIPG xenograft. But remarkably, when we look at the tumors to which these mice ultimately succumb, they are exclusively made of DIPG cells that have silenced or no longer express the dominant negative GluA2 subunit, suggesting that there is an in vivo selective advantage for cells that can engage in glutamatergic signaling. And so what I've told you is that gliomas integrate into neural circuits. They do this through neuron to glioma synapses, as well as electrically through potassium-evoked currents that are then amplified through gap junctional coupling between the glioma cells. We can begin to think about targeting these ion channels and neurotransmitter receptors in pediatric high-grade gliomas. And some early preclinical work in the lab suggests that this may be a really promising strategy. So blocking AMPA receptor signaling or blocking gap junctional function both decrease DIPG proliferation and growth in vivo. So I want to conclude with the idea that the communication between neurons and glioma cells is not unidirectional. This is a bidirectional conversation that the cells are having. So just as neuronal activity stimulates glioma growth both through secreted paracrine factors as well as through direct electrochemical communication, so too does the glioma cells increase cortical and neuronal excitability. This has been really well described in adult glioblastoma by Harold Sontheimer and Ben Deneen. We've looked at DIPG and we find that there's similarly neuronal hyperexcitability induced by DIPG, at least in preclinical models. We wanted to know whether there was, this hyperexcitability in patients, and so we collaborated with a really wonderful, brilliant neurosurgeon up at UCSF, just our neighbor to the north, named Dr. Sean Hervey-Jumper, who very regularly does intraoperative electrophysiology prior to tumor resection. So then focusing on, he's an adult neurosurgeon, so we focused on adult glioblastoma patients. We found that if he performed intraoperative electrocorticography in patients prior to their first resection of IDH wild type adult glioblastoma, recording from tumor infiltrated brain as well as more normal appearing brain around the tumor, that indeed in the tumor infiltrating brain, there's pretty stark neuronal hyperexcitability as measured by something called the high gamma band power. And so the mechanistic parallels that are evident between normal and malignant neuron glioma interactions really underscores the extent to which these diseases are hijacking mechanisms of normal development and plasticity in the childhood nervous system, and demand that we approach these cancers from a neuroscience perspective. I want to, in my very last slide, conclude with the idea that the neuroscience of cancer is not specific to brain cancer, that in many other tissue types, the nervous system is increasingly recognized to play a critical role. And this has been very well shown in a number of different adult cancers like gastric cancer, colon cancer, pancreatic cancer, prostate cancer. It's understudied in other pediatric malignancies. And I think we need to begin to approach the neuroscience of childhood cancer in a really focused way. There are a number of potential therapeutic targets that may be discovered. And these, neuroscience, mechanisms, neural cancer interactions include, not only the kind of electrical synaptic communication that I just showed for pediatric brain cancer, but also a number of different paracrine interactions in the microenvironment, as well as systemic interactions. So there's, this is very understudied, but there's clearly communication between peripheral tumors and brain, between plays a really important third part in these systemic interactions that really need to be better understood. And then, of course, as in any consideration of the neuroscience of cancer, we can't forget about the effects that our therapies exert on the nervous system and how those neurotoxic effects actually may ultimately be playing into our therapeutic responses. So many people to highlight and thank. I want to thank our collaborators, our funding sources, the people in my lab who, of course, who did this work, and not last but not least, the patients and families whose donation of tumor tissue enables all of the work that we do. Hopefully we have some time for questions now.
That was an absolutely spectacular talk, Michelle. Thank you so much. We do have some questions that are coming in through the chat, and so I'm just going to read from the questions from the audience here. The first one is, do you know the mechanism by which methotrexate leads to microglial activation? You're on mute, Michelle.
Good. Am I back? I told you I do that at least once a day. I'm sorry about that. I'm not sure. I didn't know that I went on mute. So that's a great question. What we know is that methotrexate directly activates microglia. If we take microglia in vitro and expose them to methotrexate, they assume the same sort of state of activation that we find in vivo. And then those microglia activate neurotoxic astrocyte reactivity and disrupt myelination, I think, at many levels, but in part by influencing neuronal expression of BDNF. What we don't yet know is how methotrexate and other chemotherapeutic agents activate microglia. So it's been observed in a number of different chemotherapy agents that there's microglial activation. Microglial activation is also very clearly induced by cranial radiation, and we don't know the mechanism. But what's fascinating is that the microglial activation is persistent. So it's not only when the drug is around, but for many, many, many months afterwards in preclinical models and at autopsy, we see microglial activation in patients who have received cancer therapy persistently. I will note that I kind of presented that work just to stay in time and in cartoon form. But importantly, it's not all microglia that are activated. It's quite specifically white matter microglia. And we now know from really beautiful single cell transcriptomic work from Beth Stevens' lab and others that there are many different subpopulations of microglia. They're complex cells that have really pleiotropic roles in the brain. But the axon tract microglia or the white matter microglia are a unique type that seem to be more reactive to systemic insults of a variety of sorts. So we're trying to better understand this. But if we deplete microglia after methotrexate exposure, that actually normalizes the interactions between the other glial cell types and neurons and rescues cognition.
Fantastic. So the next question is for axon glial synapse. Have you done MR spectroscopy? And if so, what metabolites were found?
Yeah, that's a great question. We have not done MR spectroscopy. We are thinking about trying to add in MR spectroscopy just to monitor neurotransmitters in the tumor microenvironment over time, but we haven't actually done it yet. So we'd love to talk more about that. It's an important non-invasive technique that might give us a lot of really insightful information.
Great. So just a reminder to everyone, you can type your questions into the chat for the panel. So I wanted to ask Michelle for the neuroligin 3 knockout mice that were used as an implantation background. Eventually, after a very impressive delay, eventually those tumors start to grow. What's the compensatory mechanism there? Are they is that going through some sort of synaptic? Do they have an enhanced synaptic activity in the tumors? Is there something else driving some of the more cancer pathways that can also be triggered by neuroligin 3?
Yeah. So we're trying to answer that question. They're very long experiments because we have to wait for the tumors to evolve resistance over those months. And so we're collaborating with Mario Suva's group, and we're isolating those tumors that have or have not evolved resistance in the neuroligin 3 knockout background, and then doing single cell transcriptomics to try to understand exactly the answer to that question. So that samples are currently being sequenced. It was a little bit delayed by the COVID crisis, but hopefully the next time we talk, I'll have an answer to that.
Great. Very good. So I'm just checking to make sure I'm not missing some of these questions coming in. So your TrkB conditional knockout in the oligodendrocytes clearly has a difference in terms of response to stimulation. What about just the basal development on those? If you get rid of TrkB in OPCs, is there abnormal myelination or anything overtly obvious, or is it really all activity triggered?
Yeah, so there definitely is a role for BDNF to TrkB signaling in normal myelin development. And so we know from work out of Simon Murray's group in Australia that, BDNF heterozygous mice or OPC-specific TrkB knockout mice both have abnormal myelin development. We were careful actually to delete TrkB after myelin develops in the mouse, and so we didn't first induce the TrkB loss until after P35.
Okay.
Yeah, but no, it's a great. So the inducible component of that was really important to sort out, to sort of separate development from adaptive myelination. And we think there's this developmental phase of myelination that we think is influenced by neuronal activity, and then after developmental myelination has completed, and it completes in mice at a developmentally earlier time point than in humans, then there is ongoing adaptive changes.
Good, got it. Okay, it says a question from Lindsay Schwartz, who says, your studies where in vivo optogenetic stimulation in the cortex influence glioma growth. Were the growth effects observed only near the stimulus site or also more distally along the circuit, for example, in regions where the cortical neurons project to?
Right. So it was throughout the circuits. We actually measured it in the corpus callosum. We stimulated in the cortex and measured the tumor growth in the corpus callosum.
Very good. Do you think what do you think the role is for the structure of the circuits in the neighborhood of the tumor for potentially driving tumor invasion or expansion?
Yeah. The role of activity-regulated invasion is a really interesting one. There's some hints coming out in the literature that there may be activity-regulated mechanisms of invasion as well. This is much less well-studied. We have some unpublished work showing that activity-regulated secreted factors do promote invasion, in experimental models, but we're still working to sort out the details of what the mechanisms really are. So it's a great question. I think that, it is clearly true that the tumor cells are integrating into circuits. And so in order to get there and integrate, there has to be invasion, whether there is, a sort of trophic component to the activity or not is suggested, I think, by these sort of, circuit patterns of spread that we sometimes see. But I think there's a lot to be studied there.
Cool. Okay, so I see we are a little bit past the hour. That went by quickly. It was a really wonderful seminar, Michelle. Thank you so much. So Michelle is able to stay a little bit longer if there are additional questions, but I know many people have scheduled an hour for the talk. So I'd just like to thank you again for a really fantastic seminar. Thank everybody for attending. And I wanted to mention that this is the penultimate talk of our Science of Childhood Cancer series. So we have one additional seminar next week, and we may have a brief slide with the title for that. But we encourage everyone to come for our final seminar next week as well. And please feel free to stay longer and continue to put your questions into the chat. So if you would like to have a longer discussion. Thank you, everyone. All right. Thank you very much, Andrew. So our final lecture for the series will be next Thursday, Dr. Eric Chow on late effects of targeted molecular therapies. All right. Okay. So here's another question. This is from Younggu Han. How does membrane depolarization increase proliferation of the tumor cells?
So I love that everyone is asking the questions that we are actively working on. It's great. It makes me think we're doing the right things. So the voltage-sensitive mechanisms of proliferation are not clear for either normal precursor cells or for the malignant precursor cells. So we're trying right now, I have a very focused project trying to understand these voltage-sensitive mechanisms of proliferation. And, I could hypothesize based on voltage-sensitive, signaling in neurons that, pathways that have been elucidated in neurons, that it's likely, dependent upon, voltage gated ion channel opening and subsequent, like voltage gated calcium channel opening and subsequent downstream effectors, but we haven't figured this out yet. So more to come there.
Yeah. Very good. I know for your neuroligin 3 studies, you had some clear connections to PI3 kinase AKT signaling coming downstream. So what proportion of the activity there is mediated through that PI3K-mTOR axis, and how much would be independent of that?
So in vitro, if we block PI3 kinase mTOR, that strongly decreases the proliferation in response to neuroligin and in response to the conditioned medium. So I think it's a really important signal, at least in vitro. But PI3 kinase mTOR inhibitors haven't been nearly as strong for us as say, Atom 10 inhibitors in vivo. And so I don't know if that speaks to the, pharmacokinetics and dynamics of the medicines we've used, but it doesn't seem to capture all of the important growth promoting aspects of this in vivo.
Right.
Yeah. And Susie, I have to apologize that my favorite video didn't work. I've never had that movie fail me.
Yeah, the movies are spectacular. I'm sorry too that didn't.
I've never had them not play.
Right. Yes. Yeah. So if you look at your xenograft growths at different sort of stages. So it's not always easy to follow a very early implantation for a xenograft. It takes a little bit of time to establish, but there is a pretty prolonged period of time where, you can start off with a really small nexus and then you start to get that diffuse infiltration.
Yeah.
Do you see, how quickly do these synapses form when you put the xenografts in there?
Yeah, that's a great question. I think because we were still establishing that they were present, we looked 4 to 8 weeks later. So we haven't ever looked immediately after xenografting, but looking at the way that these interactions evolve over time is something that we're beginning to do in a focused way. There's a graduate student who's very interested in kind of tumor evolution over time. So that's a question, again, that I hope to be able to answer. There are many questions about what regulates this kind of circuit assembly between the tumor and the normal brain. If there is any kind of circuit refinement, if microglia are functioning to prune synapses here the way they do in the normal brain or not because of influences of the tumor on microglial function, and how it evolves over time, I think is going to be fascinating. So both from the perspective of kind of serial EM to look at different, kind of degrees of synaptic integration, early versus late, and as well as we're planning to do some in vivo chronic calcium imaging to look at the way that the circuit dynamics evolve within the tumor and within the normal brain circuitry. So my student who's working on this is actually presenting a very detailed plan on exactly this question to his quals committee next week. So just whatever. Yes, definitely answers to come. That's exactly to do.
Terrific.
Yeah.
Terrific. Okay, we have another one from Lindsay Schwartz. Says, I think you mentioned 5 to 10% of patched cells had current in your recording experiments in hippocampus. Do you think that means overall that a small subset of cells received the direct innervation, or is that a low number due to technical challenges like finding connected pairs?
Yeah. So to some extent, both. I think part of it is, we found 5 to 10% of cells responded to the exact axons that we stimulated. And so that might not, really reflect all of the synapses that are present there. And I should note that those experiments were done blocking GABAergic signaling, so really isolating glutamatergic signaling in the synapse. We've subsequently, are we still recording, by the way, or have we stopped recording?
I don't know the answer to that.
We're still recording, but we could stop, I think.
Okay, I'm going to tell you about a big unpublished result, so maybe we can stop recording.
Okay. Hang on, give Andrew a second.